Okay, hi everyone. So I want to draw the attention back to the textbook. I've been doing a lot of out of the textbook stuff. So I remember when I was a student, I would always get so annoyed when the professors wouldn't follow the textbook because you had me buy the textbook, you want me to read the textbook, but what you're telling me isn't in the textbook. So I like to stick to whatever is in the book just to help you out and help you focus your time studying. So I'm looking at like page 780 and 781 right now. Which is the beginning of like the immune section. And you'll see the chart that's on page 780. This basically goes through a lot of the things that I went through in my diagrams, the different types of cell-to-cell messengers and what they do. Then you see that classical pathway for the complement on 781. Remember I told you, you really just need to know that it's you know, ultimately activating that C3b at the end. To get to that part where the cell membranes are destroyed. On page 782, you find a chart that has all of the different types of cells. We've done that already on a graphic organizer, so that's kind of checked off of our list of things to cover. And then you see the inflammatory response on 784, a nice little flow chart there. I did not diagram that out. Maybe if you're unfamiliar with the inflammatory response, it's pretty easy, pretty straightforward. You want to take some time, maybe put each of those things on a note card and see if you can mix them up and put them in the correct order. That's the way I like to do flow charts. Each event gets its own note card, maybe just a word on one side and then a description on the other side, and see if you can mix them up and put them in the correct order. On 785, you'll see the different comparisons between innate and adaptive, and that's pretty much where we left off. With the difference between innate and adaptive immunity. So now we're getting into the actual immune response and how cells recognize each other as foreign invaders and foreign antigens and kind of what they do in a response. So this is going to be a pretty big graphic organizer, and it's basically going to span the next, I don't know, maybe 5 or so pages out of your textbook there. So got my little whiteboard going, and I'll move this a little bit to get a better angle, make sure my notebook's here so I don't mess anything up. Alright, so this is the immune response, and it's going to get really small on here, but I'll make sure to go slow and spell everything out for you. And the benefit of this being a video is you could always pause it, get caught up, rewind it, etc. Do things rewind anymore? No, go back, whatever. I'm so old. Alright, so there's two types of antigen-presenting cells. There's one that's called a class 1 MHC, or major histocompatibility complex, or a class 2. And the difference between them is what those cells are. So a class 1 MHC is basically a body, a tissue cell. Maybe like your epithelial lining in your throat, right? You're getting a sore throat, something like that. A non-immune system cell. So this would be any type of tissue cell that's in your body. Whereas a class 2 MHC, major histocompatibility complex, it talks about it in your book, this would be an immune system cell, or a phagocyte, right? So these two types of cells are classified differently because they respond differently to different antigens. So the idea with these tissue cells, what they do is they will basically have a special type of T cell called a CD8 T cell that binds to it. And again, here to read everything out of your book. I'm trying to simplify it so you understand the big picture, because this stuff gets really confusing. This is high advanced physiology. There are entire master's degrees dedicated to just this chart that I'm going to draw out, just so you know. So your tissue cells will bind to a special cytotoxic T cell called a CD8 T cell. And what happens is something called co-stimulation. Co-stimulation is basically where the CD8 T cell and the cell that's been infected check each other and say, okay, you're definitely infected, right? Yeah, I'm definitely infected. Okay, I'm going to help you. Are you sure you're infected? Yep, I'm definitely infected. So this co-stimulation takes place just to make sure that this CD8 cell isn't just binding and attaching to any random cell to destroy it. So that's what co-stimulation is. It's kind of like cross-checking. With co-stimulation, now there's going to be a mitotic process that takes place. Mitosis takes place. This mitosis will produce a memory T cell and a cytotoxic T cell. So mitosis of the CD8 cell after co-stimulation, mitosis occurs, memory T cell, cytotoxic T cell is created. And ultimately, that cytotoxic T cell will destroy the target cell. So this poor little tissue cell up here was invaded by a virus, that cold virus that's going around or whatever, and this whole process takes place. Cytotoxic T cell destroys it. Memory T cell will remain floating around so that if this antigen presents itself again, this memory T cell, all it has to do is create another cytotoxic T cell to kill it. So this memory cell will then divide to create a cytotoxic T cell to destroy that antigen. It's a little bit more complicated over here with the class 2. With class 2, you have a CD4 T cell, and it's just a different type of T cell. It's just another cytotoxic T cell that binds to the class 2. And you have co-stimulation again. You cool, I'm cool. You're infected? Yes, I'm infected. Please kill me. Okay, co-stimulation is that cross-checking that occurs. What this is going to do is we're going to have mitosis again, and you're going to get another memory T cell, and a T helper cell, a TH cell created. So this T helper cell is going to cause cytokines. Remember, cytokines are just little cell-to-cell messengers to be released. And what do those cytokines do? Well, they basically help put a target on this cell. So it's basically sending up the flare saying, whoa boy, we have some immune system cells here that have been infected. We need to call in some extra reinforcements, some extra troops, so to speak. So that's what those cytokines are going to do. The cytokines are released by the T helper cells. So those T helper cells are going to call for, and the cytokines are going to call forth cytotoxic T cells, TC cells. These are going to destroy the target cell. So they're ultimately going to destroy this phagocytic cell that has been invaded. However, since it is a phagocyte that's been invaded, we want to make sure that there's even more precautions in place. Because after all, our immune system protects us. And in this case of a class 2 MHC, it's our immune system that's being attacked. So the extra steps that take place are what make this a pretty complex process. So this TC cell will destroy that target cell. Again, it's all about destroying that cell membrane, and all of these parts are kind of floating everywhere. So basically all of those antigens are floating everywhere. These helper T cells also stimulate B cells to migrate. B cells migrate to the area. Remember I said it's kind of like calling in new troops, calling in backup reinforcements, so those B cells migrate. The antigens bind to the B cell, and it's what we call sensitized. So now this B cell, I'm running out of room. So now that B cell is kind of aware that these antigens are there and wants to make sure these antigens don't do any more damage. So we say it's sensitized. Now our T helper cell comes back into play. Remember our T helper cell that stimulated those cytokines before? Sorry, I'm just kind of holding this midair. So those T helper cells will bind to the B cell and activate it, and mitosis occurs. Mitosis occurs, and what's created are memory B cells, and I'm going to go up here just because I'm running out of room down there, memory B cells and plasma cells. These plasma cells should sound familiar, right? Plasma cells will then create antibodies. And what do antibodies do? Antibodies are going to float around in your blood, and they're going to look for that particular antigen that they were sensitized to, and they're going to see around that infected cell and destroy it on the spot. So again, there's a little bit of extra precaution with the class 2 because we're talking about the actual immune system cells that are in place. So I'm going to stop this video here, and then we'll continue with the immune system in the next video.